and Michal for this opportunity to present this work here, which is actually not only my work, of course, but the work especially by Malawi, a PhD student in my group, a former PhD student who is currently about to start a postdoc in Turkey. And I also want to highlight, before I start, that this work has been done at the Max Planck Institute for Evolutionary Biology in Plön, but my group has since basically this year moved to the University of Hamburg. So I'm actually now speaking from Hamburg. And we will of course continue this kind of work. And the talk by Emily has been an excellent setting for framing for this talk here, where I want to talk about the evolution of copy number variation in the MHC, but at intra-specific copy number variations within species. And as you can imagine from this picture, I will be talking about MHC and sticklebacks. So the framework of where we see our research is, of course, in the context of host-parasite interactions, and specifically host-parasite coevolution, where it's not necessarily the direct reciprocal adaptive interactions, but rather hosts being exposed to many different pathogens and parasites having to infect at least one host, sometimes multiple host species. And the dynamics that arise from these kind of interactions and evolutionary dynamics. And the question that we're addressing in this project here is the question whether this kind of host-parasite coevolution can also drive structural genomic variation, not just genetic type, but also structural genomic diversity. And I'm just showing this here, although we've already heard a few talks about MHC, but for those who do not work on MHC, I think it's often still sort of a puzzling thing, and only seeing some weird terms or some genes doesn't tell the full story and the full potential of why we are studying MHC. So I want to briefly show its function actually. And so here you see this. Let's see if that works. The MHC molecule in red expressed on a cell surface of, for example, an antigen presenting cell. This would now be an MHC class 2 molecule, where it presents short peptides from pathogens from parasites as antigens, and these are then surveyed by T cells. So this process of MHC presenting a peptide, T cell recognizing this peptide is crucial for the whole function of MHC and its importance in immunity. And then if it's a foreign peptide and the T cell is triggered, basically then this can, for example, trigger an antibody response to the specific pathogen. And then at this molecular level, we can picture basically a mock MHC molecule variant that can bind a certain antigen, to evolve this antigen to obtain a different shape, which then leads to basically the MHC not being able to bind this anymore, and that puts pressure on the host side to evolve the MHC molecule in order to be able to bind this peptide again, this antigen again. And then you can think of these as endless circles or various ways in which this is going. So that's how we basically picture coevolution at the MHC antigen interface. A few mechanisms have already been proposed and described that are thought to select for this diversity that we see at the MHC. And one of the earliest mechanisms and probably best appreciated mechanisms is heterozygote advantage, where you simply knowing that MHC genes are dominantly expressed. An individual can be homozygous, which means it has only one variant of the MHC here, this variant A, encoded by allele A, that can bind a certain range of antigens. And if an individual is heterozygous, has two different versions of this MHC molecule, then can bind more different antigens. And this very easily put is expected to lead to better pathogen resistance. Binding more peptides means higher likelihood to recognize the peptides and trigger a specific immune response, and that usually is advantageous. So there is selection for diversity, and indeed the MHC is, as has already been pointed out, one of the most genetically diverse regions in the vertebrate genome. The human species is best studied, and if you look at the population level, we see, for example, for the MHC class 1 gene HLA-B, more than 5,000 known alleles segregating in human populations. There's a very significant sequence divergence that can be found among some of these alleles, and there are multiple MHC loci per genome usually, and we just heard about this across the different songbird species. But also at the individual level, you can have variation in the number of MHC alleles, and we saw this here. This is one of the publications from Emily that shows an overview of basically the variation in number of alleles that can be found by genotyping different individuals from different songbird species. And you see both the range of different species in which they differ in the number of alleles, but you also see within the species quite some variation in the number of alleles. And there are basically two possible explanations for this. And we actually see the same in sticklebacks. So this is a plot only for 3-spine sticklebacks, where we see for MHC class 2 genes as well, where we see a range from 1 to about 6 alleles per individual. And this can be explained in two ways. Either there is allele sharing among the loci, so that multiple loci carry the same allele. And we then, by genotyping, we see this allele only once, or by actual copy number variation of these genes within the species. And that's something we've been studying in the stickleback, because we do actually know from humans that copy number variation exists for at least for MHC class 2 genes. So these are 5 alternative haplotypes of the human MHC class 2 region, where each of the haplotypes carries a DRB1 gene, but then there are other gene copies. Some of these are functional, some of these are non-functional that carry additional DRB copies, ancient copies. And then depending on which haplotypes an individual carries, which combination, it has different numbers of MHC class 2B alleles. However, in humans, we know very little about the evolutionary relevance of this. And but we have a very similar phenomenon in sticklebacks, where we also have different haplotypes that are segregating in the population. We know these are haplotypes because the alleles segregate together in families, but we don't know, or at least until now, we knew nothing about the genomic background of these alleles. And again, there are two possible explanations. Either these are copy number variations or multiple same alleles carried by multiple loci. So if there is no copy number variation, basically you would get this kind of picture. Everyone has 3 loci, but some loci share the same allele. And admittedly, that's less likely, it's less parsimonious. So the expectation was that there is really copy number variation, but we wanted to aim to establish this at the genomic level. And for this, we went back to a what is now probably considered an old school technique. We produced back libraries, bacterial artificial chromosome libraries, and I will speed up now. And we sequenced them with PacBio sequencing, 2 of them that have 3 copies and 1 copy respectively. And we see sequence homology between the 2 haplotypes here shown in red, but we also see this stretch here of an insertion that carries 2 gene copies of the MHC2. We can look at this in a dot plot where we also see this copy homology. And we place this on the chromosome, on the genome of stickleback chromosome 7. We identified two regions adjacent to each other. One is the non-classical MHC region that carries a locus, MHC class 2 locus that is present in all individuals, which is conserved, something we also know from other species as non-classical MHC genes. And we have the classical MHC region that carries these different polymorphic loci with different copy number. And so again, we have the reference genome and the 2 haplotypes that we have sequenced here. And if we annotate the other genes, we see that, interestingly, the structural variation, the copy number variation, is really only seen for the MHC genes. The rest of the neighboring genes is largely conserved in its structure. So there's really only this copy number variation in the MHC. And if we look more closely, we identify transposable elements in a very interesting pattern, both in the reference haplotype as well in these two haplotypes that we sequenced, and also in another haplotype that was previously published, but covers only really the classical MHC region. We see that in all haplotypes that have more than one pair of these MHC2 gene, AP genes, there is a transposable element between the copies, which possibly facilitates non-homologous crossing over, as shown here. So when in a case where we have 3 loci with the transposable elements, it becomes more likely that there is non-homologous joining and crossing over, which then leads to a variation in the copy number as a result. So the presence of these TEs might actually facilitate the evolution of this kind of copy number variation. And the presence of TEs has also been shown already in other MHC regions and other species. So we see this distinct haplotypes, and the question is now how adaptive is this? How relevant is this in evolutionary terms? And we also know that there are stickleback ecotypes that we find in both northern Germany, but also in other places. Specifically in northern Germany, we see a lake and a river ecotype that are known from previous work to be adapted to local parasite communities. And what we see is that they exhibit, we genotype them, they exhibit a non-random distribution of MHC haplotypes with different copy numbers. So I'm showing here on the y-axis the copy number of the MHC haplotype, the number of MHC haplotypes, on the x-axis the number of gene copies on these haplotypes. You see that in river fish, we see more often haplotypes with a single locus, and in lake fish, we see more often haplotypes with 3 or 4 gene copies, which indeed leads then to a difference in the individual number of MHC alleles. For river fish, we see on average about 3 alleles. For lake fish, we see on average about 3.5 to 4 alleles. And this might suggest that there are different optima in the individual MHC diversity between these two habitats because of different parasite communities. And indeed, the parasite diversity is lower in the rivers, and which makes it likely that this difference in MHC copy number is also adaptive in this context. So what we find here is that the variation of the individual MHC allele numbers results from interspecific copy number variation, that this evolution of this might be facilitated by transposable elements, and that this may be adaptive in the context of local parasite communities. With this, I will end. Thank the people in my group, but especially Malawi for doing most of this work, also a number of collaborators, and highlight again that we've now moved to Hamburg in northern Germany, where we start the research unit for evolutionary immunogenomics. With this, I thank you and look forward to any questions.